वेलकम टू टुडेज क्लास। आज का जो टॉपिक है वो है न्यूक्लियस। सो न्यूक्लियस, इफ दिस इज अ सेल, then inside the cell nucleus is present. So each cell contains cytoplasm. This is cytoplasm and this is nucleus. So each cell has nucleus, and nucleus is very important part of cell. So we will study structure of nucleus. So this is the diagram of nucleus. Nucleus is bounded by double membrane, and this double membrane is called as nuclear envelope. So this is nuclear envelope. So nucleus is bounded by double membrane and it is called as nuclear envelope. So this is nuclear envelope. So this is nuclear envelope. Nuclear envelope has small pores, and these pores are called as nucleopores. These pores are called as, see, this is pore, this is also pore, this is also pore which is present in the nuclear envelope, and this pore is called as, all these pores are called as nucleopores. Nucleopores, singular number, nucleopore. Inside the nucleus, nucleoplasm is present. So this is nucleoplasm. This is nucleoplasm. Nucleoplasm. And nucleus, in nucleoplasm, one dark body is present, and this dark body which is present inside the nucleus, in nucleoplasm, this dark body is called as nucleolus. So this is nucleolus. This is nucleolus. And this nucleolus, function of nucleolus is production of ribosomal protein and RNA. Function of nucleolus is production of ribosomal protein and RNA. Then this nucleolus contains chromatin material. So this is chromatin material. Thread-like structure is present inside the nucleus, and this thread-like structure which is present inside nucleus is called as chromatin material. This is called as chromatin material or chromatin fiber. Nuclear envelope is decorated by ribosomes. So these are ribosomes. These are ribosomes. Nuclear envelope is decorated by ribosomes. So these are ribosomes. Ribosomes. And this nuclear envelope, it is connected to endoplasmic reticulum. This is endoplasmic reticulum, like this. So this nuclear envelope is connected to endoplasmic reticulum. It is connected to endoplasmic reticulum. Here also we can draw endoplasmic reticulum. So this is endoplasmic reticulum. Then after completion of diagram, we will discuss functions of each part of nucleus. So this is diagram of nucleus. Very simple. Nucleus is bounded by double membrane, and these double membranes are called as two membranes collectively called as nuclear envelope. This nuclear envelope interrupt at some places to form small pores, and these pores are called as nucleopores. Nucleus is filled with material, and that material is called as nucleoplasm. This material which is present inside the nucleus is called as nucleoplasm. Here is the label nucleoplasm. Nucleoplasm contains one dark spot, one dark body, and this dark body is called as nucleolus. And the nucleoplasm contains thread-like material, and this thread-like material is called as chromatin material. Nuclear envelope is decorated by ribosomes. So see, these ribosomes are attached on the external surface or on the outer surface of nuclear envelope, and nuclear envelope is connected to endoplasmic reticulum. So this is endoplasmic reticulum. Then we will discuss functions of each part of nucleus. So first part is nuclear envelope. Nuclear envelope. So first of all we will discuss the structure and then function. So nucleus is bounded by double layered membrane called nuclear envelope. It is called a nuclear envelope. Then nuclear envelope has pores called nucleopores. So nuclear envelope is the boundary of nucleus. So this is eukaryotic nucleus. In the prokaryotic nucleus, nuclear envelope is absent, and in eukaryotic nucleus, nuclear envelope is present. So this nuclear envelope is the boundary of nucleus, and it is present only in the eukaryotic nucleus. Nucleopores regulate the movement of material from nucleoplasm to cytoplasm and from cytoplasm to nucleoplasm. See, this is cell, and this is cell, and this is nucleus. Nucleus contains nucleoplasm, so this is nucleoplasm, and outside the nucleus cytoplasm is present. So this is cytoplasm. This is cytoplasm which is present outside the nucleus, and nucleus is filled with nucleoplasm. So this is nucleoplasm. So these are nuclear pores or nucleopores. Function of nucleopores is nucleopores regulate the flow of nucleopores regulate the flow of material from cytoplasm to nucleoplasm and from nucleoplasm to cytoplasm. So nucleopores control the movement of material or flow of material from cytoplasm to nucleoplasm and from nucleoplasm to cytoplasm. So material from cytoplasm enter into the nucleoplasm through nucleopore, and material from nucleoplasm moves out in the cytoplasm through nucleopores. So nucleopores, function of nucleopore. Nucleopore regulate flow of material from nucleoplasm to cytoplasm, nucleoplasm to cytoplasm, and cytoplasm to nucleoplasm, and cytoplasm to nucleoplasm. So please write down all these points. Next, Nucleolus. It is a large rounded body. Present in the nucleoplasm. So the shape of nucleolus is differ from cell to cell. For example, in human egg, that is human female gamete, nucleolus is very large. Nucleolus is large in size. This is very important point. According to NEET, in human egg, nucleolus is very large in size, and this nucleolus in case of human egg is a different name. It is called as germinal spot. In case of human egg, this nucleolus is very large in size, and nucleolus in human egg is called as germinal spot. Next point. But in case of human sperm, nucleolus is very small in size. Nucleolus is inconspicuous. Inconspicuous means small in size. It is very small in size, and it is very difficult to locate inside the cell. Means it is inconspicuous. Next point. So please note down all these points. Function of nucleolus. Synthesis of RNA. Synthesis of rRNA, that is ribosomal RNA, and ribosomal proteins. So nucleolus, function of nucleolus is that synthesis of ribosomal RNA and ribosomal proteins. So ribosomal RNA and ribosomal protein synthesized inside the nucleolus, and then so rRNA and ribosomal protein synthesized differently. Means if this is rRNA, it is synthesized by nucleolus, and this is ribosomal protein. If this is ribosomal protein, this is rRNA and this is ribosomal protein synthesized inside the nucleoplasm by nucleolus, and then this rRNA and this ribosomal protein move from nucleoplasm to the cytoplasm. So if this is cell and this is nucleus and this is nucleolus. So synthesis of rRNA, that is ribosomal RNA and ribosomal protein, takes place inside the nucleus by this nucleolus. Then this ribosomal protein and ribosomal RNA, this is ribosomal protein and this is ribosomal RNA, move out from this nucleoplasm and enter in the cytoplasm, and then they assemble. Assembled means they bind to each other to form ribosome like this. So this is ribosome, and in the ribosome rRNA is present. So this ribosomal protein and rRNA produced separately inside the nucleus by nucleolus. Then rRNA and ribosomal protein moves out, enter into the cytoplasm and assemble together to form ribosome. Next point, that is nucleoplasm. Nucleoplasm contains nucleolus, then chromatin material and some organic and inorganic substances. So main thing is chromatin material, which is present in the nucleolus. Chromatin material appear like this thread-like structure. It is thread-like structure. This is chromatin material. But this chromatin material appear thread-like in non-dividing cell. Chromatin material appear like a thread. Means when cell is in interface, when any cell is in interface, interface means it is resting phase of the cell, then in that cell chromatin material is appear like this. But when that cell undergo division, that is mitosis or meiosis, any division, when that cell undergo division, mitosis or meiosis, chromatin material condensed and formation of chromosome takes place. So chromatin material. Chromatin is made up of DNA, that is deoxyribonucleic acid, and histone and non-histone proteins. So chromatin material is made up of DNA, deoxyribonucleic acid, and histone and non-histone protein. So when any cell undergo division may be mitosis or meiosis, any division. So when cell is in metaphase. Metaphase, when cell is in metaphase. Metaphase is the one of the stage of cell division, that is mitosis. We will explain it. See, if this is cell with nucleus, if this cell undergo mitosis, it is a cell division in which chromosome number remain constant, and mitosis has two stages, that is karyokinesis. Karyokinesis means division of nucleus. Karyokinesis means division of nucleus. And cytokinesis. Cytokinesis means division of cytoplasm. Division of cytoplasm. So, this is cell, and inside the cell, this is nucleus. When this cell undergo mitosis, see, after completion of karyokinesis, single nucleus divide into two nuclei. So, division of nucleus is called as karyokinesis, but when cytoplasm divide, so division of cytoplasm is called as cytokinesis. In this way, single cell divide into two cells. So, such cell division, so this process is called as a cell division. And cell division, there are two types of cell division, mitosis and meiosis. So, that may be mitosis or meiosis, any type of cell division. First step will be karyokinesis, that is division of nucleus, and second step will be cytokinesis, that is division of cytoplasm. So, in case of mitosis, karyokinesis takes place in four steps, and that steps are prophase, second one is metaphase, third one is anaphase, and last is telophase. So during metaphase, see, this is cell, and this cell is in metaphase. Metaphase is going on in this cell. Then spindle fibers. These are spindle fibers. Nuclear envelope is disappear, and chromosomes appear. So see, these are chromosomes. These are chromosomes. Chromosomes appear in metaphase stage of cell division. So this is the diagram of metaphase, and in metaphase chromosomes are clearly seen inside the cell. So why I am drawing this diagram? To clear the difference between chromatin and chromosome. So this is chromatin material which is appear inside the nucleoplasm, inside the nucleus, when cell is in resting phase. That is when cell is in interphase. But when cell undergo any division, that is mitosis or meiosis, these chromatin fibers condense, become short, become thick, and chromosomes form. So these are chromosomes. So these are chromosomes. So formation of chromosomes takes place from this chromatin material. This is chromatin material and these are chromosomes. So when chromosomes are formed from chromatin material? Chromosomes are formed from chromatin material when cell is in division stage, exactly in metaphase stage. So this cell is in metaphase stage. So in metaphase stage, chromosomes are clearly seen. Chromosomes are arranged on equatorial plate of cell. So these are chromosomes. Chromosomes are made up of DNA and histone. And DNA, that is deoxyribonucleic acid. DNA is genetic material. DNA is hereditary material which carry genetic information from one generation to next generation. So therefore, this nucleus contains chromatin material. Chromatin material is made up of DNA and histone, and DNA is genetic material. So that's why this nucleus in cell is very important part. So nucleus is very important part of cell. So in this way we have discussed all about the nucleus.